There are two major types of false positives: similarity-based and function-based. False positives due to similarity can be because of remote matches or due to near neighbors between highly pathogenic agents and those that aren't as severely pathogenic or not pathogenic at all. Some common reasons for this are inappropriate thresholds or very short sequences, which can cause spurious alignments, or sometimes the sequences themselves can be highly repetitive, leading to the wrong identification. False positives can also be caused by incorrect assessments of function. A sequence from a highly pathogenic agent may still be safe if it's related to housekeeping function, for example, DNA repair or a ribosomal protein, or if the function or sequence are conserved across different types of organisms, some of which may be non-pathogenic or some of which may be less severely pathogenic. In the next few slides, we'll go through a few examples showing how this may show up in DNA screening. Here we have an example of a sequence that identifies as deriving from anthrax, but the gene is a DNA-directed RNA polymerase. DNA-directed RNA polymerases are enzymes that help with synthesizing RNA from a DNA template and can oftentimes be found in lots of different organisms. And so in this case, while we have a match to anthrax, we also have a gene that's typically considered housekeeping. In this next example, we have a match to anthrax and also matches to other related organisms within the Bacillus family, like Bacillus cereus. While some of them are pathogens and may cause disease, they're not nearly as severe as Bacillus anthracis, and so don't typically correspond. These genes that are shared amongst them wouldn't typically correspond with what makes Bacillus anthracis much more pathogenic than, for example, Bacillus cereus. And so in this case, with given the best match being to anthrax and some of its relatives, this is an indication that it may be conserved and that the gene is probably not responsible for the main reason that anthrax is highly pathogenic.